Natterson-Horowitz here with us today. So she is a cardiologist and evolutionary biologist on the faculty of Harvard Medical School, Harvard University's Department of Human Evolutionary Biology, and the School of Medicine at UCLA. Her phylogeny lab studies the natural world as a source of insights to improve health and development. One focus of her research has been identifying evolved adaptations in other species as a source of insights for human cardiovascular challenges, from HFpEF to atherosclerosis to cardiomyopathy and atrial fibrillation. Her New York Times bestseller, Subiquity, co-authored with Catherine Bowers, was a finalist in the American Association for the Advancement of Science Excellence in Science Books Award, a Smithsonian Top Book of 2012, and a Discover Magazine Best Book of the Year. Widhood, a New York Times Best Book Review Editor's Pick and winner of the Association of Science Education Book of the Year Award, chronicles the transition from juvenile to adult biology and behavior across vertebrate species. Her recently launched initiative, Mental Health Across the Tree of Life, focuses on comparative psychopathology as a source of insights for human mental health. So without further ado, here we have Dr. Natterson-Horowitz.
All right, great to be here. And I want to introduce, before I start, my co-presenter, Dr. Karam Gill. And we've been working together now for a few months, and he is going to be presenting a really interesting project looking at arrhythmia suppression. The potential that electrogenic fish may represent a natural animal model for arrhythmia resistance. Just FYI, he's a hospitalist here starting in October. He's applying in cardiology, and he just finished his residency at All of You. And a lot of people, I think, know him. He's wonderful. All right. So I'm going to talk quickly because we have all kinds of cool stuff, and I want to make sure that Karam has a chance to really feature his cool work. So all of my work in the past 15 years is based on kind of a single idea that our field of medicine is quite anthrocentric, human-centered. And apart from a handful of model organisms that we use in labs to understand human health, we really we disregard the other hundreds of millions at a minimum of metazoan species. But if we could expand the window through which we perceive health and disease to include those other extant species with whom we share the earth, looking at commonalities and differences, and if we could then again expand that window even farther to take a look at the evolutionary history as depicted by this phylogeny, the evolutionary history that shapes those similarities and differences. If we could do that, we could, number one, do a better job at understanding why, the causes of illness, and we could be more effective, more nimble, more creative at developing biomedical innovations to counter disease. Well, those are a couple of big claims. I have, I think, maybe 30 minutes or so before Karam's going to go on. So here's what I'm going to do. A whistle-stop tour through my journey from clinical cardiology, heart failure echocardiography here for many, many years. Introduce you to kind of thinking about disease from a phylogenetic perspective, a very, very short overview, and then give you one example that I use frequently to show how a phylogenetic perspective can give us insight into causation that we might not otherwise have. And then really lean into what we're going to talk about today, which is looking at the evolved adaptations of other species as a roadmap for innovation in cardiovascular medicine, and particularly in arrhythmia, and particularly in sudden death. All right, so my journey is basically trained here in internal medicine, in cardiology, in my fellowship, did advanced training in imaging and heart failure, and was happily working as a cardiologist here. In the mid-2000s, LA Zoo asked if I would become a cardiovascular consultant for their patients. First, it was great apes, and then it expanded beyond that. And basically, over many years, I became very, very interested in the shared diseases of human and non-human animals, particularly the shared non-communicable diseases, because many of us have a good sense that animal and human health intersect at the level of zoonoses. But what about non-communicable diseases? What about cardiovascular disease, cancer, psychiatric disease? And so that became really quite a focus and led to a significant shift in my life and career. But of course, I'm certainly not the first physician to recognize that there is a real connection and opportunity. Rudolf Virchow, one of the founders of modern medicine and certainly modern pathology, wrote in the 19th century, between animal and human medicine, there is no dividing line. nor should there be. The object is different, but the experience obtained constitutes the basis of all medicine. And remember, he's a pathologist, so he's looking through a microscope at cells. He is looking past the fur and the feather and the fins, the distracting elements that perhaps have prevented others from recognizing these connections. In the past, now almost 15 years, I've been publishing books, creating conferences, courses, programs at multiple levels in the US and internationally, and embarking on research agendas, including this phylogeny lab, where I've had wonderful experience over the last 5 years or so with people at primarily medical students and graduate students, but sometimes we have a range of others who participate. All right. So back to this question, how could I use way back then all of this gained knowledge about the spontaneous occurrence of so-called human non-communicable diseases in other species to improve human health? I'm a physician after all. And since I'm a cardiologist, today we're going to look at what I was thinking about then, and focusing on how can we use this broad comparative phylogenetic, as I'll explain, knowledge to improve human cardiovascular health. Well, what we do is we need to apply an evolutionary lens. And by that, I mean a phylogenetic lens. Here's a very, very brief history, just to get everybody up to speed. Okay, so we're going to start with the Big Bang, 14 billion years ago. The Earth comes online about 4.5 billion years ago. You saw the moon up there. And we're not, of course, going to see life until 3.6 billion years ago, when we have single-celled organisms, aquatic only. Photosynthesis comes online. You're going to see the little oxygen bubbles. And ultimately, of course, the Earth is saturated with oxygen with one of the great extinction events, the Great Oxidation Event. But ultimately, we see by the time we're at about 600 million years or so, we are looking at multicellular life and the first metazoans, real animal life after this glaciation event. And of course, we then are going to be heading directly toward, that's right, the Cambrian explosion, where, of course, we saw this incredible diversification of species, the terrestrialization of species, and so on and so forth. I think people are familiar with this. There's a bit of a lag, I see, on the Zoom, but we'll get there. All right, so what I want you to do is just for a moment, train your eyes right up here. That's a timestamp, right? There's the meteor. So let's say that's 65 million years. Here is our common ancestor with chimpanzees, right? That's about 6 to 7 million years. And of course, you don't see humans here. Why? Because this is 6 to 7 million years. This is 65. Our species is 200,000 years old. And remembering that evolution works by descent with modification, which means the solution, the phenotype, uses the biology that preceded it, creates a good enough solution. And so we know this from genomics, but it's important to kind of, I think as physicians, we can get so focused on the human that we can forget just how much we share with other species. Just to put it in one other context, if you think about the history of life and the history of the Earth as an airplane ride from LAX to New York City, it would go something like this. The Earth started about 4.5 billion years ago, right? So we're going to see first life sort of over New Mexico, Arizona. We're going to see photosynthesis over kind of Colorado. But it's not going to be until we end up in Western Pennsylvania that we have a kind of multiplasticity and multicellularity that allows for metazoans, for animal life to evolve. And so that this is sort of a 600 million year timeline. It's kind of fun to see what's happening during this period. We're going to come upon the age of dinosaurs, the extinction of dinosaurs. We're going to look at the common ancestor of chimps and humans. And then there we are, 570 feet from touchdown in New York City, our species evolved. So much more commonality than difference, but centuries, millennia of human exceptionalism perhaps have created a kind of blindfold that we need to kind of periodically be reminded of our status as animals and this common ancestry. So let's talk about phylogeny, and let's take Pennsylvania, why not, and create an x-axis. Phylogenies come in different shapes and forms, but this one, the x-axis, is the timeline 600 million years ago and the present. And phylogenies are what I use. I have, over the last decade or so, learned a lot about how to build these models. But of course, what are they? They're family trees that evolutionary biologists use to depict the relationship between species and species characteristics. This, of course, is probably the most famous of all phylogenies. It's the only illustration in The Origin of Species. One of the things I absolutely love about it is this. Talk about humility. That's just the greatest ever note to self. So what I do is I'm thinking about a trait of vulnerability to disease. That is the trait that I'm going to build phylogenies for. Well, let's take a beat, because we're physicians here, and ask, what is vulnerability? Well, from a physician's perspective, when we talk about differential vulnerability, we're really talking about the differences in vulnerability between our patients, all of whom are Homo sapiens. So this is an intra-specific conceptualization of vulnerability, right? On the other hand, if you are thinking from an evolutionary perspective, phylogenetic perspective, then vulnerability can be cast, and differences in vulnerability, as a species-specific trait, and one would expect it, in different phenotypes. There's multiple forms of phenotypes at multiple levels. So we're going to talk about interspecific, not intraspecific, but interspecific vulnerability. During the pandemic, PNAS published it was a paper looking at vulnerability to COVID, right, across a number of species. And I sometimes use this just to remind people that this is that vulnerability to a disease, which is going to be a complex trait with multiple interdependent pathways and lots of biology. It's an evolutionary trait that we can map onto a phylogeny. And in fact, when we see phenotypic differences, on a phylogeny, all of a sudden it invites us to look to explain why these differences in this trait exist. And of course, the answer can be found in the evolutionary history that has shaped these different phenotypes. We'll get back to that. And so I could do systematic reviews. And for many years, this is all I was doing, was really finding cases of so-called human diseases and building models to depict how ancient and how widespread vulnerability to and various non-communicable diseases are. All right, so I promise that I'm going to give you a quick example of how a phylogenetic perspective can enhance our understanding of causation. And so, hey, let's use a really common cardiovascular issue, vasovagal syncope. Now, a lot of you probably, maybe on rounds this morning, you had some medical students who asked, why does this happen? And you explain the mechanism of let's talk about like a fear-induced kind of a syncopal episode, right? Sort of a classic, where there's this sympathetic activation leading to the activation of the ventricular afferents and the sympathetic withdrawal, the vasodilation, and then, of course, the legendary bradycardia, which when I, many years ago, taught second-year pathophysiology course, I would call paradoxical, because I was taught that it was paradoxical. Maybe some of you have heard that it's paradoxical, that bradycardia. Why? Because what is the prediction with fear? You expect fight, flight, right? So this is the running of the bulls. But of course, that's not what happens. We see the opposite. Well, if you are an anthropocentric cardiologist, then maybe it's acceptable to say that it's paradoxical. But if you want to be a species spanning and evolutionarily informed cardiologist, then you might be interested that in one of the first of the studies that I did, I found examples of fear bradycardia, not occasionally, right, in species, in basically in every vertebrate lineage. And so when you see a conserved trait, it is a clue about causation. So let's just go ahead and map out. And remember, the tips of this phylogeny, this one's a little bit different. On this type of phylogeny, the outside is the present, right? Over there is present, and this is the common ancestor at 3.5 billion. And so when you see a trait at the tips, these are all extant species, right? One hypothesis is that that trait existed in the common ancestor. And generally, when you see a conserved trait, and there are exceptions to this, it suggests that there may be biological benefit. Put in evolutionary terms, fitness advantage, survival advantage, repro advantage, etc. Okay, all of this should be pretty common sense. Well, this allows us to, once we map that, to sort of develop a new hypothesis. And a hypothesis, which, by the way, is a testable hypothesis, not a just so story, which is what my undergraduate thesis advisor was Stephen Jay Gould, who was a very famous historian of science and evolutionary biologist. And he cautioned against coming up with evolutionary hypotheses that you couldn't test. Well, these are testable hypotheses. So just really quickly, many underwater predators hunt using electroreception. They have anatomy, these spicules that allow them to do that. And of course, as you would imagine, if you're hunting, your signal is something like this. If all of a sudden there's a very profound bradycardia, it creates a kind of acoustic blindness, and that might be advantageous to a advantageous to a prey species. Some of you may have aquaria. You know that when you clean the tank of an Oscar fish very often. This happens. And there's lots and lots of evidence of this. This was a study in 1979. There was some fawn. They put telemetry leads on the fawn, and their heart rates were 180 beats per minute at baseline. They had mounted speakers on Jeeps, and they played recordings of wolf howls, right? This is 1979, and to frighten them. The fawn, and they saw this plummeting of the heart rate down to 30 beats per minute. And then this is the associated physiology. And you can begin to imagine why that might be adaptive. And in fact, yes, as an anti-predation strategy, stillness, tonic immobility is very effective. Very quickly, in the moments before what I'm about to show you, there's a young cheetah that had taken down an impala. A mature hyena has sort of bullied in and has the feed, but the cheetah's about to return. So there's a spotted hyena digging in. Cheetah's like, hey, I took that animal down, and the hyena's chasing it away. Now I want you to focus on the impala, not the hyena. And among the animals where can be a very significant alarm bradycardia are African ruminants. There's more about that, but that's not the subject of today's lecture. Today we're going to look at how a phylogenetic perspective can not only teach us about causation, but can help us do a better job of innovating more, come up with more effective innovations. Okay, so just then, moments ago, I showed you this kind of what I call the outside-in approach to phylogeny, where we're looking at a trait that exists in modern extant species, and then we're looking at common ancestry, and there's a series of ideas that can be and hypotheses that we can generate. But there's another way of thinking about phylogeny, and that's to flip it, and that's what I call the inside out, which is here's the common ancestor, and of course what we are seeing as we move out is this diversification, right? This is all of these different species with all of this phenotype, all these varying phenotypes represent biodiversity. It's the process of speciation. And why does speciation occur? Why is there biodiversity? Well, simply stated, when we have animals live in an environment, and when environments change, which they do, either animals move to new environments or environments in place change, then there are going to be phenotypes that are better or worse. And the ultimate phenotype, that is the winning sort of suite of genes, and the phenotype that is best is that phenotype that is maximally aligned to that environment. That is, that can maximally counter existential threats to life, but also has phenotypic characteristics that allow it to maximally leverage opportunities and resources in the environment to maximize reproduction, right? So classic. So successful phenotypes contain evolved adaptations, right? That's what you're seeing when you look at biodiversity and speciation, that there are evolved adaptations, and those that are most aligned with environmental challenges and opportunities, those are the ones that are going to have the greatest fitness advantage. One example I think everyone here is very familiar with, you probably studied it as an undergrad, are these fish that are both in the Antarctica and Arctic that have evolved these antifreeze glycoproteins. And this is an example of convergent evolution. But obviously a fish, a tropical fish, if you got the water down to, Arctic or Antarctic water levels would the blood would freeze, would crystallize. But these fish have evolved as they have migrated towards the polar extremes, the ability to produce this antifreeze, which actually lowers the freezing point, so essentially protects them. Okay, and you can see that this is just to show that the phylogeny tracks with the poles, in essence, where the phenotype is aligned to the environment, right, when the evolved adaptations are beneficial. All right, so leveraging evolved adaptations in nature to develop a human approach or to solve a human problem, that's something that is called biomimicry. Many examples of this. You are probably very familiar at this point with biomimicry. The classic story is the Shinkansen trains. These are the bullet trains that were introduced in 1964. It was a very big moment. It really was a signal that Japan was recovering after the war. And it was very successful with the exception of one thing. The trains, when they decelerated into the station and decelerated out of the station, created a lot of vibration, reverberation, noise, and damage. But fortunately, one of the engineers was an amateur ornithologist. He was a weekend birder, and he knew that the kingfisher kingfishers are known for these extremely long and sleek beaks that can pierce the water without introducing any vibration, any rippling, any reverberation. And what this evolved adaptation does is it allows the animal to leverage the opportunities in the environment to access food, right? To not scare away the prey. And so Aichi Nakatsu redesigned the train, and it is a very famous story. Well, let's talk about biomedical solutions, and then let's really focus on cardiovascular. And what I do, and I this keeps changing depending on what the projects are, and is basically I'm interested in identifying and then leveraging evolved adaptations in other species which appear to have solved some physiologic challenge. In other words, that appear to confer a level of resistance to some human pathology, and to then characterize the biological mechanisms that underlie this putative resistance. Hypothermia, you just saw with the fish. This was a recent Frontiers in Science paper where there are many species now that are being looked at for the potential that may have evolved adaptations to solve a number of human health challenges. Mechanisms exist now for cancer resistance in the bowhead whale, Brandt's bat, and of course, many of you are familiar with the African elephant cancer suppression story, Peto's paradox. And it turns out that African elephants have 20 copies of tumor suppressor gene P53. And it's a longer story than that. That's been part of a very piece of a story that's evolving. And I learned how to make a QR code and put it on my deck. So I thought instead of showing slides, but I realized you don't have your phone out. If you want more examples from different papers, I'm happy to share them. But let's talk about cardiovascular issues, and let's talk about HFpEF. Why not? So I think everybody here knows, I don't really need to do too much explaining what happens with high blood pressure, and we have this thickening, and of course we're looking at, in a sense, it's a Laplace's law kind of a situation. But when the thickening occurs, there's of course changes to the muscle, there's hypertrophy, fibrosis, and there's structural and functional changes that lead to, all of the things that we know, the observed changes in the histopathological changes, the hemodynamic changes, and of course the clinical changes. We have patients who are short of breath, fatigue, whose exercise tolerance is not good. Well, if I were trying to find a species that might have evolved adaptations that solve this challenge of HFpEF, I decided that I would turn to the giraffe. And why the giraffe? Simply because the giraffe has the highest blood pressure of any animal. And that is because the left ventricle is about here, and about almost 3 meters vertically is the brain that needs to be perfused. It's a longer story than this, but that's the sort of quick and dirty on it. And why the long neck? Many theories you're probably exposed to as a child. The idea that, the giraffe can access foliage and food, and scanning the horizon. If you are living on the savannah and predators are a leading cause of death, having a long neck gives you that advantage. And a long, but I think now refuted hypothesis of sexual selection. But I can't bear to give up that slide. But I don't think sexual selection is actually a selective advantage. But let's go back to this high blood pressure. Why? Well, it's Laplace's law, and it's not high blood pressure, and it's not hypertrophy. There's nothing pathological about it. The giraffe has a very thick ventricle, but despite a ventricle that looks like that, and if you think about what would happen in a human patient who had long-standing systemic hypertension, nice LV, significant hypertrophy, and all of that, you'd have a symptomatic patient. But the giraffe has this blood pressure, compared to human, very thick, and yet every day flees easily at least 40 kilometers an hour of predators. So clearly something's up. So I found this was probably about 6 or 7 years ago. I first started just staring at this and thinking, what the heck? And a number of things happened that allowed me to kind of get into this question. But I had a fibrosis suppression hypothesis because we had looked at a bunch of. Anyway, the long and the short of it is, here's the hypothesis. We've got only 2 surviving extant members of Giraffidae: giraffe, the modern giraffe species, and the okapi. And here's the okapi. You may recognize them with the interesting striped legs. And here are the giraffe species. Well, the common ancestor lived about 11.5 million years ago. And the ancestors of the okapi moved into the Congolese rainforest, where a lengthened neck would really not confer adaptive advantage whereas the ancestors of the modern giraffe species, of course, moved to the savanna where it would. And so the hypothesis was that over the course of that 11.5 million years or so, evolved adaptations emerged to confer the resistance I'll show you in just a moment. And these are ancestral species of giraffe. I work with a couple of really marvelous scientific illustrators. And for my Scientific American feature, Dino Puleri, who did this, he did all of that artwork. And so there's Olu, and we went to Denmark and we did some of these experiments. I also started saying yes to invitations to image okapi. You can see the iconic legs there. And we were about to, no, I'm sorry, I had just submitted this first paper when Chang Lui and his group published this fantastic paper, in which they looked at, called a towering genome, and they basically looked at giraffe, okapi, and other ruminants, and they did genomic comparisons. And bottom line was they found certain unique genes in giraffe, including FGFR1. And what they did was to take groups of mice. They had wild type, they had the experimental group. They CRISPRed giraffe FGFR1 into the experimental group, and then they exposed both groups to 28 days of angiotensin II, reduced hypertension. And what did they find? A few things, including, here is the giraffe angiotensin II exposed. Look at that in terms of fibrosis versus the wild type angiotensin II. And in fact, this is the fibrosis in the wild type versus fibrosis. So fibrosis suppression is maybe part of the story. It's not the singular explanation, but part of it. There are many other stories like this, and I'm working to do that with all kinds of groups. I don't have a traditional lab. I typically get someone interested in something, and then I'm atypical, but it's endlessly interesting, and when people understand it, they often want to participate. So I mentioned a few moments ago that the field of human medicine was very anthrocentric, but it's also been, not a big surprise, androcentric. And for that reason, I've been sort of focusing for the last several years on female health. And so this was the PNAS Nexus paper that looked at evolved adaptations in species that confer resistance to high impact issues in women's health, from reproductive senescence, which is a big challenge, osteoporosis, etc. Okay. So what time is it? Early in terms of
12:30. We're good.
Oh, good. Okay, excellent. I just want to leave lots of time for my fantastic partner here. So, you know, in thinking about, first of all, of course, HFpEF is not uniquely female problem, but we also know that, you know, we understand that it is a significant issue in women's health, and so I would consider that part of this female health across the tree of life initiative. But the 2019 Nobel Conference was, the theme of it was bio-inspired medicine, and I was invited to keynote, and I was invited to invite people from around the world who I thought might be, you know, who would have something to say about this topic. And so I invited the giraffe folks, and that was wonderful because we all hung out for a week and just learned and learned and learned, and that's how some of these collaborations happened. But Christian Alcager, who's a Danish physiologist and really one of the leaders in this, he was showing his data, and he was all the giraffe stuff, and I asked, not to be provocative, but I just asked as well, how many of the giraffes were male and how many were female? And he said, well, actually none of them were female. And you know, I think all of us probably, you know, hear that and are not surprised for all kinds of reasons. They weren't discriminating against the female giraffes. There were some very solid reasons that was the case. But even so, I started thinking about the issue of pregnancy and the issue of gestational hypertension. Because if giraffes evolved adaptations to protect their myocardia from the effects of hypertension, then one would expect that there might also be evolved adaptations protecting mom and fetus from the adverse effects of blood pressure elevation. So this became, that started another project with some fetal and maternal health folks at UC San Diego, and that's ongoing, and there's lots of, it's a matter of bandwidth and people who are interested in moving it forward. So, but in any event, lots of other questions and ideas. I had a Harvard undergrad who did her thesis, she did a thesis or a senior paper on pandas and osteoporosis, because pandas are one of, you know, female mammals ovulate. For us, it's 12 times a year, let's say. But other animals, it's different. There's ovulation is its own issue. Pandas ovulate once a year. Once a year. There's one 72-hour window in which they can fertilize an egg, and that can be fertilized. So what that means, think about it from a bone health perspective. You can imagine a human who had only one ovulatory cycle and didn't have estrogen exposure. It would be not hard to imagine. And so there was her hypothesis was that there were evolved adaptations relative that were related to bamboo consumption because of the phytoestrogens in bamboo. That's not been tested, but again, you understand how this works. We know that the panda has very strong femur bone. The Chinese have done extensive research on this beloved species, and so we have a lot of information. So that's the kind of project. Also, really interesting, reproductive senescence. Female in our species, right, is a very big issue. A lot of emphasis on it right now. And the Greenland shark is a viviparous shark. That is, they conceive. They have a placenta. They actually, that's right, a shark placenta. And then they produce pups, live birth. That's not unique to Greenland sharks. Many sharks do that, but Greenland sharks are extremely long-lived. For the book on animal puberty, and I learned that they go into puberty at about 140 years of age. That is, the females start to ovulate at 140, and then they're fertile for maybe 150 or 200 years. That means they're ovulating, conceiving, gestating healthy litters for hundreds of years. And so to me, that sort of, yes, it's a shark, it's not a mammal, but still it's a vertebrate, and that is intriguing. Could there be a blueprint there for expanding reproductive length in our species? Okay, what is the road to the bedside right now? Well, the road to the bedside is it's a road, right? We're not there yet. I went a little bit nuts on the QR codes. Apologies. There are 3 companies that are. I'm sorry. The first 2 are companies, and the third is the Monarch Initiative. But if you're interested, Peel Therapeutics is Peel and Fauna. Peel, Josh Shipman started. Josh is the oncologist who did the elephant P53 study with Carl O'Malley. Fauna Bio, a former student, anyway, Ashley Zender started Fauna Bio, and if you're interested, so those may be of interest to you. The road to the bedside it exists. It is. I'm not going to say it's long or short. I think that is to be determined. There are a lot of people interested in this, and there's a lot of money that's going into seeing what can be found. I'm going to end in just a moment, but I would like to before I do so, just give you a couple of quotes that I really like. I have a few scientific heroes. Rachel Carson is one of them, not just because she launched the modern environmental movement, which she did with the publication of Silent Spring in '62, but she really understood the connection across species. In nature, she wrote, "Nothing exists alone." And with apologies to her, I would say the Female Health Project and all of these projects, whether we're looking at developmental similarity across species, disease vulnerability based on sex, in nature, no female exists alone. So there is this continuity that links animals, not human and non-human animals, a continuity of shared vulnerability to disease, but also this idea that there can be adaptations that confer special resistance to some other species, and that protecting that kind of biodiversity can be very valuable, not just for that species, but for us. Finally, I would say that in a moment, you're going to hear a fascinating project that Karam Gill is engaged in, looking at the evolved adaptations in electrogenic fish that may confer resistance to sudden death, to VTVF. And if you think about all of the money and energy that's gone into research and development and where we are, now think about evolution, this hyper-iterative, massive scale R&D kind of project, right? Where the phenotypes that work are selected and those that don't aren't. Imagine that, and then you'll understand how prescient it was when Charles Darwin wrote in The Origin of Species, "Natural selection is a power incessantly ready for action and," wait for it, this is the best, "immeasurably superior to man's feeble efforts." So with that, I want to introduce Karam, and he will share his project with you. I'm going to go off. No, I'll stay on share, yeah. And we'll do questions at the end.
Gets me every time.
He's using my laptop, which he doesn't like the mouse pad. There we go.
Looks like we got the slides up here. Yeah. So we want to introduce you to one of our other projects, something that we're really excited about. And once again, show you how we would like to apply this phylogenetic approach. I like to call it the Natterson-Horowitz approach to another sudden cardiac death. The sudden cardiac death represents a significant burden of mortality within the United States. And typically the terminal arrhythmia is ventricular fibrillation. But to say that VF causes SCD would be a pretty vast oversimplification. In fact, the development of VF and sudden cardiac death is predicated on a multitude of factors, like a patient's anatomic or functional substrate, acute changes in the cardiac milieu, things that create heterogeneity between cardiac myocytes and predispose them to arrhythmia mechanisms such as cell-cell coupling and reentry. And then all it takes is one spark, set things off, something like your classic RNT phenomenon. But the question is, how much of a spark is required to cause VF in a human? And there's actually a group of cardiothoracic surgeons in Australia who looked at this question. And what they did was they measured the minimum current required to induce VF at different levels of the heart. And what you can see is kind of what you would expect. The farther out you move, the more current is required. Note here, the minimum fibrillatory current at the level of the epicardium was 735 micro. But what about from an external stimulus? And we can actually refer to our friends, the electricians over at OSHA and their safety guidelines. And what we see is that at currents as low as 50 milliamps, there is a chance of inducing VF. And then when you get to currents greater than 1 amp, it carries a significant risk of VF and death. To put this into context, your typical household outlet carries 15 amps of current. And then for those curious, a lightning strike carries a massive 30,000 amps of current. We do actually have ECG data from lightning strike victims in the field, and 50% of the time they have VT or VF. But as we talked about, the propensity for VF varies across individual humans. But let's take that step back, let's take that Natterson-Horowitz approach and ask ourselves, do we see that variation in propensity for VF across species? And you guessed it, yes, we do. We actually have data from multiple different species that show varying thresholds for VF. I think one of the more interesting cases is that of the American alligator. The American alligator, being cold-blooded, is exposed to a variety of low body temperatures, right? It's got to use the sun to get its temperature up. And so a group of scientists hypothesized that because it's exposed to these low body temperatures, it will have some evolved adaptation that will allow it to have a higher threshold for VF, given that hypothermia can incite VF. And so they tested this. They took alligators' hearts, they placed them in 23 degrees Celsius and 38 degrees Celsius, and they paced them at different rates. And they compared them to a very metabolically active animal, the rabbit. Now, in this experiment, the rabbits did not fare so well. All the rabbits developed ventricular fibrillation at 23 degrees Celsius and even at 38 degrees Celsius when paced rapidly enough. However, under no circumstances throughout the experiment did an alligator develop VF. Another one of my favorite cases is that of three-toed sloth, probably the most metabolically inert mammal that you can think of. A group of Brazilian scientists seeing this evaluated the minimum threshold or the minimum current required to induce VF in sloths, and they compared that to similarly sized dogs. And what they saw was that it required a significant amount of current, significantly more current in the sloths to induce VF. But I think more interestingly, 40% of the sloths actually had spontaneous reversal of their VF within 60 seconds of onset. Now, there's one group of relatively unstudied animals that we thought provided an opportunity on the topic of sudden cardiac death, and those are the electrogenic fish. The electrogenic fish are fish that have developed the capability to utilize electricity for a variety of purposes, and they've done this over millions of years. Those purposes being navigation, self-defense, hunting. And there are 26,000 or more species of teleost fish. These are your bony fish, but very few fish that can actually use electricity. And they developed this capability convergently and across continents. You can see the majority of these electrogenic fish are located in either the South American continent or African continent. However, you can find them around the world. So what we believe is that prey species of these electrogenic fish engaged in this evolutionary arms race with pressure of electricity will have over time developed adaptations to allow for a higher threshold for ventricular fibrillation. And furthermore, these electrogenic fish, being the most exposed to this electric current, will have evolved for life with a higher VF threshold as well. So what do we know at this time? We know that these electrogenic fish are capable of releasing high voltage discharges, these volleys of electricity during hunting. And you can see an electric eel demonstrating this capability here as it preys on this fish, the top right of your screen showing that discharge. The electric eel is probably the most well-known electrogenic fish, probably the most well-studied, and it can release about 860 volts of electricity in household outlets. That's 8 times your household outlet, and that is a lot of electricity. If you don't believe me, we got a video here to show you. This is a video of an electric eel who is caught on a fishing line, and an unfortunate caiman seeing this said, hey, I got an easy meal. But you can see what happens when the caiman takes a bite out of the electric eel. This rigidity as the electricity flows through it occurs, and then after some time, the eel passes, the electricity wears off, and then you can see, though, that the alligators succumb to all this electricity that's flowing through it. Now, the assumption is that the main inhibitory mechanism used by electrogenic fish during predation is myotomy. However, we believe, given what we know about the threshold required to induce VF, that it's equally likely that a ventricular arrhythmia is at play here. And why is that important? Well, this might be a little bit older. Let's see. Slides here. See if we can go through.
Canva is great until it is, yeah.
There we go. Jump forward 20 slides. That's okay. And so, like I was saying, why is this important? Using that phylogenetic approach, that Natterson-Horowitz approach, we can go ahead and identify these evolved adaptations that these species have, leverage these adaptations for biomedical innovation. You can see there are a plethora of possibilities that could be utilized for something like human electrophysiology here and VF suppression. So how do we go about doing this? I'd like to introduce you to our model organism, the torpedo ray. This ray, living in a salty ocean conductive environment, is capable of producing vast amounts of electric current. Up to 30 amps is hypothesized at times. Fun fact, the torpedo, the underwater missile, was named after these fish, actually. And though they're not as well studied as your electric eel, it does confer to us one specific advantage. They live right off our coast, and they have a whole plethora of prey species as well that are subjected to its electrical currents. So the torpedo ray produces its current via 2 electric organs that sit on either side of its head, noted here, these large blue pulsating ovals on the top of your screen. And what I really want you to note on this slide is that red circle. That is the relative location of the torpedo ray heart. And so you can see its proximity to these highly electric organs, and it really begs the question, what mechanisms, what methods does this ray have to protect itself from electric accidents? Some possible hypothetical mechanisms here may be that this ray has developed some sort of protective anatomy, some specialized tissues that confers increased resistance to electric current. We heard from Dr. Natterson-Horowitz that fish at times use increased vagal tone in lieu of a flight or fight response, and so it's possible that by lowering its heart rate, it's not detected by electroreceptive predators. Could that same mechanism be at play here and leading to protection against VF? Another thought is channel adaptations. These electrogenic fish develop their electrical capabilities through loss of function of a sodium channel in their muscle cells over millions of years. It is not far-fetched to think that it's possible that they have other adaptations as well to their channels that help to protect them against VF. So going forward, we are in the process of obtaining Pacific torpedo array specimens. Once we have those specimens, we can characterize their anatomy and electrophysiology. And if our hypothesis holds true, then we can identify the unique genetic adaptations that these fish have that are not present in their non-electrogenic relatives. Very similar to the giraffe and the okapi. But we are kind of embarking on this fresh journey with tons of possibilities. And so we thought one of the best ways to go ahead and move forward also would be to acquaint ourselves with our electrogenic fish, its prey, and its ecosystem. And so our next step is also to hit the water. We're going to be renting a dive boat, going out just off of Long Beach. And so for all my scuba divers, it doesn't sound like there are any scuba divers in the room, but maybe in Zoom, you can come out and dive with us and see if we can't find these fish. If you are not a diver, Dr. Natterson-Horowitz will be hanging out on the boat. She's not a diver either. You can come join us for a wonderful afternoon on the ocean. If you are interested, you can go ahead and email me. My email should pop up on UCLA MedNet. But in the meantime, we are happy to take any questions.
So as we wait for questions in the chat, I guess for both of you, I'm a little curious with these big mammalian projects, what ethical things do you come across because some of these animals are extinct? Or like, how do you know if you're fishing and like know that they've already, procreated, and then which kind of like what are some of the
they were kind of our plan in kind of general terms is to go diving and not to catch the fish. Oh, that's more like a social. I wanted to say, I was like, oh no, no, we're not fishing. But the projects, many of the projects that I do are done basically at this point using publicly available bioinformatic data. I mean, just comparative genomics, working with just different bioinformatics specialists to kind of come up with hypotheses. Now, in the case of some of the other stuff, the giraffe, there are groups around the world who have IACUCs that are done through their countries. In the case of the Danes, and so no one is doing anything that is, if anything, I mean, one of the pitches that I make is that all of us I know feel that we should support preserving biodiversity for all of the moral reasons. Our species has had far too great of a destructive footprint. But if there are people who don't have that kind of a commitment, just knowing that I will accept their support of protecting biodiversity, even if the beneficiary is still human. And I think there's a very strong case that this project makes that there may be evolved adaptations that are solutions to challenges that we have deemed intractable. So there's the human interest piece of it. There are changes afoot that are, I think, really exciting. So I've been a member. So the National Academies has the Division of Earth and Life Sciences, and for decades, the only board within the National Academies has been ILAR, the Institute for Lab Animal Research. But about 5 years ago, which was a directive from the executive director, was to expand that, to say, hey, there is now research that involves animals, conservation research, wildlife research, One Health, comparative oncology, clinical trials in animals. There are these domains that are far outside of the lab. And so in October next month, ILAR is ending, and the National Academies is launching a new board, which is the Board on Animal Health Science, Conservation, and Research, at which I'm co-chairing, and which will be having conversations about ethical questions, among other things. One of which is there's a huge amount of research that is done on animals, and research in clinical trials, which is research on humans. Mm-hmm. We need to find ways to, of course, protect subjects and welfare, but also to direct the beneficiaries of that research needs to include other species. Mm-hmm. We need to, even if we're human investigators, it's an MD PhD, it doesn't have to be a DVM PhD, that it's a moral imperative, especially for a 21st century physician, given what we know about planetary health, to be considering what species can benefit other than our own. So there are opportunities to think in an ethical way, but to have an expanded ethical conversation.
And from someone who's fresher in this field, my perspective on it, I was actually very surprised by a lot of the information that's missing within the veterinary field. And one of the things that we do is we try and connect both human and veterinary medicine. So a good example of this is the Great Apes Health Project, which Dr. Natterson-Horowitz introduced a little bit of, but we go to the LA Zoo, and there are other zoos around the country that we perform TTEs on great apes, and we're trying to create a standardized measures that can be used to analyze these TTEs, because they don't really exist at this point. If you think about when you're trying to do a TTE on a gorilla, so much more goes into it than when trying to do it on bedside in the emergency department. So we just don't have as much data. So connecting veterinary and human medicine is something that we hope comes out of all this research.
Feel free to put any questions in the chat. Do we have any other questions in the room here?
So, how many volts did the electric field fire up?
860.
And that was enough to take out the crocodile.
Yeah.
Is that your hypothesis?
I think so. Yeah. If the 860 volts can generate a large amount of current. It varies on the resistance, but in the medium. But you can see that came in there. It did not thrive after it had received
but
that
even
degree of
with
electricity.
The threshold,
Exactly.
There's a limit to, of course, that you can take out anybody.
Right. So there's you can get the whole.
That's clearer than what the evolutionary idea that you could apply to human is.
So a couple, good, very good question. So first of all, remarkably, when you look at the numbers, which is what we're trying to make the case, the amount of voltage that the amount of electricity that is discharged is sufficient to take out, assuming human-sized and individual horses have been taken down by electric eels, for example. So, but despite that, the entire literature about the mechanism through which electrolytic fish hunters are hunting is that the electricity creates like myotony, right? Myotonic, it's all muscular. And I actually think the reason, it's just what I encounter very often, is no one thought of VT, or if they didn't publish on it, if they did think about it. I think they were just not, it happens, right? So there isn't any, but it seems to me as likely and possibly even more likely, or at least as likely, that inducing arrhythmia which would have the same behavioral effect, the functional effect, which would be to slow the prey species down. So the first thing we do not know, the first question we have to answer is what is happening from a rhythm perspective when a prey species receives a charge. But that's not your question. Your question is, what is the road to the human bedside? Yeah. So here's the basic.
Or to building in protection for humans. So
here's the way it would go. So we all are familiar with this concept of an evolutionary arms race, right? So, a prey species gets a little bit faster, and then the predator, evolves the phenotype that allows it to go a little bit faster. So you see these, you can actually match certain cardiac troponins if you look comparatively in predator and prey. So we know that one gets better, the other better get better, or they're not going to survive, and that's right. So that's the classic evolutionary arms race. We see that in antimicrobial resistance, right? The bacteria develops resistance to an antibiotic. We have to come up with a different antibiotic. They develop a resistance. It's this. That's that's what happens across evolution. Now, in the case of the electrogenic fish, my original hypothesis was, well, the animals that are surviving and thriving in that environment, they may be vulnerable, but they are probably it probably represents a phenotype that has acquired some degree of resistance, right? And therefore, because the model that I use is to say, okay, what is the human pathology that we need help with, right? Where can we find an approximation of that challenge in the natural world? So, for example, if you're trying to develop new approaches to food safety and anaerobes, you might think of going to look at the physiology of a carrion-eating species like a vulture or hyena who has evolved adaptations to safely consume pathogens that we kill, right? So you have to think of where in nature is there a solution, a problem and the solution. So that's how, so the idea is that the prey species or some of the prey species, remember that the electricity is not only going to be affecting the animal that it's directed at, right? There's going to be an effect that they would have to have some degree of protection, either at the muscular level or at the heart level. And so does that make sense? So once you've said, okay, we think that that's the case, then you have to test that. You have to see whether in fact that's the case. But if there is a resistance, then sort of in the way of the elephant P53, then you begin to look for the biological mechanism that may underlie it. And that's the concept that there may be that evolution may have produced novel approaches, solutions to these otherwise intractable issues. From the perspective of the predator, this was something that Michael Habib and Shiv and others were kind of thinking about, that, which it never occurred to me that, well, you've got these electricity generating organs basically adjacent to the heart. So maybe the first species to look at would be the one that, is presumably in greater exposure. But
internal safety mechanisms.
Well, correct.
Well, for example, you mentioned they evolved the nets of a sodium channel, making it inexcitable.
Right.
Yeah. So it was a study that was done to kind of figure out where this electrical capability came from, and they looked at different electrogenic fish, and they all in some way had lost the specific sodium channel within their non-electrogenic cells. Whether it was the loss of an enhancer or just complete loss of function. But through that pathway, they all came to the point of developing the electrical organ. So like you're saying, that's another possible method that could be at work.
The Monarch Initiative, the Broad Institute, there are mostly mammals, but the genomes of huge numbers of animals are now being sequenced much more than 5 years ago, and certainly 10 years ago. We did not find a complete sequenced genome on the torpedo, California. So we have to double, triple check that. But so, there's a lot of work to be done. But it's what our goal here is to introduce an approach that requires really thinking in a broadened way and beginning to think about how evolutionary pressure, that selective pressure, will produce solutions. When I went back to the giraffe situation, there's this moment where I looked at it and I thought, okay, there's selective advantage to having a long neck, but the cost of that is a thickened ventricle, right? Because it's a Laplace. Now, if the cost of a thickened ventricle was fibrosis and a slowed giraffe, that long neck never would have evolved. Those genes would have been taken out by a predator. So what the long neck essentially did in a way is to create an evolutionary crucible where you have the fitness advantage of a long neck, the potential liability of a thickened ventricle leading to exercise intolerance. And so the necessity for an adaptation to allow for a thick ventricle, but not to compromise exercise capacity.
We have one question from the chat actually relating to that. So it seems that they have severe hypertension, LVH, but not heart failure. Beyond understanding potential treatments, does this call into question the correct understanding of the mechanism of HFpEF?
Oh, that's interesting. And I'm going to just quibble a little bit. It's an anonymous question, so don't take it personally. So it's not, it's a little anthropocentric to call it hypertension or hypertrophy, right? Those are terms that imply pathology. So it's a fully normal, healthy adult giraffe is going to have a thickened, a thick wall to cavity size that is substantially greater than in humans and in other non-long-necked ruminants of comparable size. So there's a wonderful JEB paper that actually the group that I was with in Denmark, that they actually looked at the hemodynamics. They tried to understand what was going on. It was quite an impressive study. They used a TEE probe, threaded it through the IJ. I don't have time to show you, but if you want, I can show you what the size of the IJ on these are. And they used it as an ice catheter, and they actually did diastolic flow. They paced and they infused to create naturalistic pressures. And they did not find, what I thought they were going to find would be increased stroke volume or some, so anyway, we don't know. But, and there are some, I want to say, to be fair, there are some that would argue that maybe, anyway, they're inside the world of giraffe heart physiology. There's some debate, which if people are interested, I'm happy to share papers and talk about.
Okay, great. Well, thank you so much for coming. That was fantastic. Lots of praise from
It was fantastic.
Thank you. And please, if you're here, take a sandwich on the go. Sorry to be late, or soda or water for dinner.
Or